Hello, everyone. Thank you for joining me this afternoon. So today I'm going to share with you my Keystone project, which draws inspiration from my experiences as a competitive figure skater, and it aims to investigate how stress can impact motor memories. I've had the good fortune of being advised by Professor Steve Ramirez, whose own research focuses on memory manipulation. So first, to contextualize the phenomenon I'm researching, I imagine many of you are familiar with those scenes from horror movies where a person running for their life can't get into their locked home because they're fumbling with the key, or a critical moment in a football game when the receiver fumbles the ball, or even something as basic as struggling to tie your shoe when you're in the rush to get out the door. All of these things share in common a stressful situation in which the person experiences impairments in fine coordinated motor movements that are otherwise routine or habitual. As I mentioned previously, the specific motor impairing stress event that sparked my research idea comes from the sport of figure skating, where skilled motor movement is essential for a flawless performance. Particularly frustrating is when a skater pulls out of a jump early and does not complete the intended number of rotations, causing the jump that has been trained in practice to look like this, to end up looking in a stressful performance more like this. But it's more than just competitive athletics that have to gain from studying this phenomenon. Many high-stress occupations, including surgeons, first responders, and military personnel, rely on highly trained motor skills that often make the difference between life and death. As such, these people have to be extremely good at overcoming the potentially detrimental effects of stress in order to do their job right. In my study, we aim to use mice to model the phenomenon under investigation, but how can mice serve as an adequate proxy for human behavior? It turns out that in many ways, mice can be more useful for studying these questions, primarily due to the ability to use more invasive techniques when conducting research in mouse models versus humans. Because of these methods, we are able to investigate circuit level mechanisms and directly manipulate them in order to study the relationship to behavior. In memory, this corresponds to methods which identify and control engrams, the groups of cells that are involved in coding a particular memory. And here on the right, we can see individually fluorescently labeled neurons. And lastly, there is some degree of evolutionary conservancy between mice and humans, and the development of effective behavioral tests which model human phenomenon couple these circuit level strategies in a meaningful way to support foundational research. So my project right now currently exists as an NRSA grant proposal. Unfortunately, we were not able to conduct these experiments in the lab due to concerns surrounding COVID, but this will sort of run you through what we intend to do perhaps in the future. So it consists of 3 aims, which we will go through sequentially. The first aim seeks to identify, label, and control a motor memory. So in order to achieve this aim, we will first use established genetic techniques for effective labeling and controlling of engrams. But firstly, how do you identify an engram? The key to this question lies in immediate early genes, or IEGs, which are genes that are transcribed when a neuron is active, thus providing a reliable marker of cellular activity. Using transgenic mice, we can control the window during which our genetic construct is live so as to specifically tag neurons in the target brain region that are active during a certain episode. Here, we will tag neurons during skilled motor learning on the Rotorod task. This task essentially involves placing mice on a rotating rod that accelerates over the course of 5 minutes. The goal is to have the mice learn how to stay on the rod for longer and longer periods of time. Lastly, in order to control the neurons associated with that motor memory, we can use optogenetics, which enables the activation or deactivation of neurons using light. The experiments for this aim will be divided into 2 paradigms, one that activates a deactivated motor memory and one that inactivates a learned motor memory. In both setups, we will begin with a training period during which the mice learn the motor skill of staying on the Rotorod. The notation off dox, which is seen in orange here, indicates that we will change their diet to lack doxycycline for this portion of the experiment, which allows us to tag the neurons associated with the motor memory. When the mice are placed back on dox, this prevents neurons that are active during the rest of the experiment from being labeled and confounding the results. For the activation group, we will then inject them with a drug which induces amnesia, causing the mice to forget the motor memory. The deactivation group does not receive this drug. On the following day, we will assess the ability of the mice to successfully recall the previously learned motor memory, as well as our ability to manipulate that memory. Here, for both groups, we begin with an unstimulated trial to assess the memory on its own. For the activation group, which experiences amnesia, we expect that they will not perform well in this first trial. The deactivation group, on the other hand, has intact memory and should perform as well as when they learn the skill. From there, we will either activate or deactivate the memory tag during the learning trials using optogenetics. We will compare the trials when the light is on versus when the light is off to determine if the engram has an effect on motor memory recall. When we perform the recall trials, we expect to see the following results. For the inhibition group, when the light is on, the motor memory neurons will be turned off, and we predict to see a decrease in motor performance, showing that these cells are necessary for motor expression. Conversely, for the activation group, when the light is on, the neurons that participated in motor learning will be reactivated, and we predict to see an increase in motor performance, showing that they are sufficient to induce proper motor expression. Together, we expect these two experiments will demonstrate successful labeling of motor engrams. Our second aim seeks to investigate the ability of stress to modulate the motor engram we identified in aim 1. We seek to accomplish this by tagging a separate stress-related memory and using it to interfere with motor recall. So first, how can you stress out mice? There are actually a variety of ways to do this, the most common being loud noises, confined spaces, and bright lights. By using these three conditions simultaneously, previous research has shown that a single 1-hour event is sufficient to induce acute stress. To assess the efficacy of this setup, we will use the open field test, a behavioral test used to evaluate anxiety-like behaviors in mice. We will place the mice in an open field and measure the time they spend at the outer edges versus the center. Though mice are curious creatures and like to explore, they also tend to avoid open spaces, and so the more anxious they are, the more time they will spend at the outer edges versus the center. Lastly, we will again use the Rotorod test to assess motor recall, as well as the ability of the stressful event to interfere with motor performance. We will begin as before with the Rotorod learning trials. Next, we will assess the mice in the open field test to establish a baseline level of anxiety for each mouse. We will then change their diet temporarily to allow our genetic constructs to label active neurons. This will occur while the mice are subject to a 1-hour stress event so that the neurons we tag are associated with the stress memory. The following day, we will return the mice to the open field, this time using optogenetics to activate the neurons from the stress memory in order to behaviorally evaluate the engram's capacity to induce anxiety. Finally, we will return the mice to the Rotorod task for evaluation of recall, again simulating the stress engram to observe its effects on motor performance. Given that the neurons we labeled for control are associated with the stressful event, our prediction is that the reactivation of these neurons during the open field test will induce anxiety-like behaviors, which will translate, as I said before, to more time spent at the edge of the apparatus. We will also predict that reactivation of the same neurons will interfere with motor memory recall and thus decrease performance on the Rotorod task. Finally, our last aim seeks to investigate the mechanisms by which stress may interfere with motor memories. Specifically, we will study stress hormones as the chemical agents of stress and whether or not they can alter the neural circuits associated with motor memory. Research has demonstrated that the behavioral expression of a memory is correlated with the degree to which the same cells that were recruited during memory encoding are reactivated during memory recall. Therefore, our hypothesis is that stress and the production of the stress hormone corticosterone will result in a decreased overlap between encoding and retrieval cells, which behaviorally will manifest as decreased performance on the Rotorod. To test this, we will use a drug that blocks the action of corticosterone and observe motor output and neuron reactivation. In our experiment, we will have 3 groups. Each group will undergo motor learning, followed by either a stress or a non-stress event, finally followed by motor recall. Group 1 will be given the drug prior to recall in order to reverse the effects of the stress event, while Group 2 will not receive this drug. Group 3 will be our control, and they will experience a non-stress paradigm as seen as remaining in their home cage. At the end, the brain samples will be collected from each group and evaluated for cellular overlap. For our results, we expect to establish a baseline level of overlap with the control group, which is Group 3. Compared to this baseline, we expect to see a decrease in the number of reactivated cells in Group 2, which experiences the stress event without the drug. And lastly, when the corticosterone inhibitor is injected prior to recall, we expect to see a return to baseline levels of overlap, which indicates that the drug prevented the effects of stress on motor memory recall. It's important to note that this study is just a preliminary investigation of a much larger set of questions. So here we begin to probe neural circuits, underlying mechanisms that facilitate the stress motor memory interaction. However, more research will be needed to expand upon these mechanisms to apply them to broader situations. For example, how does repeated stress affect long-term or repetitive skilled motor memory recall? Moving beyond mechanistic studies, we also hope to eventually begin understanding why in humans, some individuals respond differently to stress, especially when it comes to performance enhancements caused by acute stress. And finally, the ultimate goal is to uncover potential strategies by which individuals who struggle with motor performance under high stress situations may overcome this hurdle. Thank you so much for listening to my presentation, and I'm happy to take any questions at this point.